1.27 million. That is how many people died last year from antibiotic-resistant infections. More than HIV, more than malaria. And there is a treatment that has worked against bacterial infections for over a century. Not a new experimental drug, not a breakthrough from some cutting-edge lab. A virus. A specific kind of virus called a bacteriophage that hunts bacteria and destroys them. The Soviet Union used phage therapy on millions of patients for decades. It worked. The West looked at the evidence, looked at where it came from, and decided to ignore it. I found this while researching the history of antibiotics. I was trying to understand a simple question. If penicillin was discovered in 1928, what were doctors using to fight infections before that? The answer, it turns out, is not nothing. The answer is bacteriophages, viruses that evolved over billions of years to do one thing: find a specific bacterium and destroy it. A French-Canadian microbiologist named Félix Dairel identified them in 1917 at the Pasteur Institute in Paris. And what happened to his discovery over the next century made me realize the antibiotic story we were all taught is missing its most important chapter. The deeper I went, the more I realized this was not a scientific failure. This was a choice. Félix Dairel did not set out to rewrite medicine. In 1917, he was investigating an outbreak of dysentery among French soldiers at the Pasteur Institute when he noticed something he could not explain. He took filtered stool samples from recovering patients, added them to bacterial cultures, and watched. The bacteria dissolved. Something invisible in that filtrate was hunting the bacteria and killing them. He called them bacteriophages, eaters of bacteria. Dairel was not cautious about what he had found. By 1919, he conducted what is believed to be the first human phage therapy trial. He treated a 12-year-old boy suffering from severe dysentery at the Hôpital des Enfants Malades in Paris. He administered the phage preparation orally. The boy recovered within days. Over the next decade, Dairel's work spread across the globe. He treated cholera outbreaks in India in the 1920s, working with the British Indian government. He traveled to Egypt, to the Soviet Union, to South America. Everywhere he went, the results were consistent. Phages targeted specific bacteria with a precision that no chemical compound could match. They replicated inside the very bacteria they were destroying, and then moved on to the next cell. A living, self-amplifying medicine. In 1923, a Georgian microbiologist named Georgia Eliava invited Dairel to Tbilisi. Together, they founded what would become the Eliava Institute of Bacteriophages, Microbiology, and Virology. The Soviet government funded it. By the 1930s, the institute was producing phage preparations on an industrial scale, treating everything from dysentery to wound infections to surgical complications. The Red Army carried phage preparations as standard medical supplies. But here is what the standard history leaves out. This was not only happening in the Soviet Union. In the United States, major pharmaceutical companies saw the commercial potential. Eli Lilly, one of the largest drug companies in America, began producing and marketing commercial phage preparations in the 1930s and 1940s. They sold phage products for staphylococcal infections, for abscesses, for upper respiratory infections. These were not fringe products. They were manufactured by a Fortune 500 company, sold to American doctors, and used on American patients. And then they stopped. In 1928, Alexander Fleming noticed a mold-killing bacteria in a petri dish. By 1943, the United States and Britain had industrialized penicillin production to treat wounded soldiers in the Second World War. The results were extraordinary. Penicillin was broad-spectrum. It killed many types of bacteria at once. It was relatively simple to mass-produce. And critically, it was a chemical compound, a molecule, something that could be standardized, bottled, patented, and sold at scale. Phages were the opposite of all of that. Each phage targets only one specific strain of bacteria, Treatment required identifying the exact pathogen first, then matching it to the right phage. That meant diagnostic work before treatment. It meant maintaining vast libraries of different phages. It meant a product that could not be standardized into a single pill for millions of patients. But what bothered me more was the economics. A broad-spectrum antibiotic is one product for 1,000 conditions. A phage is 1,000 products for 1,000 conditions. From a manufacturing perspective, from a profit perspective, the math is obvious. Antibiotics scale. Phages do not. At least not within the business model that Western pharmaceutical companies had built. Eli Lilly quietly discontinued its phage product lines in the late 1940s. The official reason was that early American phage studies had produced inconsistent results, partly because the science of phage biology was still immature, partly because some preparations were poorly made. That much is fair. But the solution to inconsistent results in any other field of medicine would be more research, better methodology, refined protocols. The solution here was abandonment. I spent weeks trying to understand why the inconsistency argument was enough to kill an entire field of research. And then I found the timing. The same decade that the West abandoned phage therapy, the Cold War began. Soviet science became, almost overnight, politically suspect in Western eyes. Soviet medical journals were not translated. Soviet clinical data was not reviewed. Soviet results were dismissed not on scientific grounds, but on ideological ones. The Iron Curtain did not just divide nations, it divided medicine. On one side, Georgia's Eliava Institute continued refining phage therapy. They built phage libraries containing thousands of preparations. They treated millions of patients. They published results in Russian language journals that almost no Western researcher read. On the other side, the West bet everything on antibiotics, and for a while, it looked like the right bet. Alexander Fleming himself warned about what was coming. In his 1945 Nobel Prize lecture, he said that there may come a time when penicillin could be bought by anyone in the shops, and the ignorant man could easily underdose himself and expose his microbes to non-lethal quantities of the drug, making them resistant. It took less than 4 years for his prediction to begin. By 1947, the first penicillin-resistant Staphylococcus aureus strains were documented in hospitals. The medical establishment responded with new antibiotics, methicillin in 1960. Then methicillin-resistant staph appeared. Vancomycin became the drug of last resort. Then vancomycin-resistant enterococcus appeared. Each generation of antibiotics bought time. Each generation of bacteria caught up. Consider the math. Between 1940 and 1962, more than 20 new classes of antibiotics were introduced. Between 1962 and 2000, only 2 new classes reached the market. The pipeline did not slow because science ran out of ideas. It slowed because the economics shifted. Developing a new antibiotic that might be used for a week costs roughly the same as developing a drug for chronic conditions that patients take for decades. Pharmaceutical companies followed the money. They moved toward drugs for cholesterol, for blood pressure, for diabetes, conditions that require a lifetime subscription. Meanwhile, the bacteria kept evolving. MRSA, C. diff, drug-resistant tuberculosis, drug-resistant gonorrhea. The World Health Organization declared antimicrobial resistance one of the top 10 global health threats facing humanity. In 2019, a study published in The Lancet estimated that 1.27 million deaths worldwide were directly attributable to antibiotic-resistant infections. Another 4.95 million deaths were associated with resistance. And while this was happening, while the death toll climbed year after year, there was an institute in Tbilisi, Georgia, that had been solving the same problem with viruses for 100 years. Their phone was not ringing. And that is when 2016 becomes impossible to ignore. In March 2016, a man named Tom Patterson, a professor at the University of California, San Diego, was dying. He had contracted a multidrug-resistant strain of Acinetobacter baumannii while traveling in Egypt. Every antibiotic his doctors tried failed. He was in septic shock. His organs were shutting down. His wife, Stephanie Strathty, an infectious disease epidemiologist, began searching for anything that could save him. She found phage therapy. Working with researchers at Texas A&M, the Navy Medical Research Center, and a team from San Diego, they identified phages that could target Patterson-specific bacterial infection. The FDA granted emergency compassionate use authorization. They administered the phages intravenously. Within 3 days, Patterson began to recover. He walked out of the hospital. Which brings me to what you're paying for right now. The average American will be prescribed antibiotics 12 times before the age of 20. The global antibiotics market was valued at over $42 billion in 2023. If your doctor prescribes you an antibiotic today, you are relying on a technology that bacteria are systematically learning to defeat. And the backup plan? The treatment that could target your specific infection with a precision antibiotics never had, was used by millions of people on the other side of a political border for the better part of a century while the West looked the other way. Phage therapy is not science fiction. It is not experimental. It is the oldest antimicrobial strategy in clinical use on Earth, and you almost certainly have never been offered it. Which brings me to the detail that does not add up. The official explanation for why the West abandoned phage therapy is reasonable at first. Early American studies in the 1930s and 1940s produced mixed results. Phage preparations were inconsistent, some were contaminated, the science was young and the methodology was sloppy. When antibiotics arrived and delivered immediate broad-spectrum results, the medical establishment made a rational decision. They went with what worked more reliably, more quickly, at greater scale. I accepted that explanation for about a week. But here is what that explanation leaves out. Eli Lilly was not a fringe operation. They were one of the largest pharmaceutical companies in the world. They manufactured phage products and sold them to American doctors. The inconsistent results were not a fundamental flaw of phage therapy. They were a manufacturing and diagnostic problem, solvable with better science. The Soviet Union solved them. Georgia solved them. The Eliava Institute refined its methods, built comprehensive phage libraries, and developed standardized preparation protocols that produced consistent clinical results across decades and millions of patients. The West did not lack the science to do the same. It lacked the economic incentive. Antibiotics were one product for 1,000 conditions. Phages were 1,000 products for 1,000 conditions. One model scales inside a pharmaceutical business structure. The other does not. This was not a failure of evidence. This was a failure of accounting. The West did not reject phage therapy because the science was bad. They rejected it because the business model was inconvenient. And then the Cold War provided the perfect cover. Soviet research could be dismissed without being read. Georgian clinical data could be ignored without being evaluated. An entire field of medicine was buried not by disproof but by disinterest, rationalized by geopolitics. The timing is precise. Antibiotics became profitable. Phage research became Soviet. And the West stopped looking. Not because the evidence said stop, because the money said stop, and the politics made it easy. I keep returning to Tom Patterson lying in that hospital bed in San Diego in 2016. A man dying of an infection that no antibiotic on earth could treat. His wife, an epidemiologist, had to discover phage therapy essentially on her own, and then convince the FDA to grant emergency authorization for a treatment that had been in continuous clinical use since 1923. Think about what that means. In 2016, the United States of America treated a bacterial infection with phage therapy as though it were an experimental last resort. In 2016, in Tbilisi, Georgia, it was Tuesday. 93 years. For 93 years, the Eliava Institute had been treating bacterial infections with phages while the West built, sold, and watched the slow failure of an antibiotic empire that Fleming himself warned would not last. 1.27 million people died of drug-resistant infections in 2019. Not because no treatment existed, because the treatment that existed did not fit the business model that Western medicine had built around patentable chemical compounds, because the country that had perfected it was on the wrong side of a line drawn on a map in 1947. Every time you take an antibiotic and hope it still works, you are paying for a decision made 80 years ago. A decision that said phages were too complex, too specific, too Soviet. A decision that said profit scales better than precision. A decision whose consequences are now killing more people than HIV and malaria combined. If this is the kind of investigation you want to see more of, subscribe. This is not the end. There are hundreds of these stories, and the pattern is always the same. A treatment that worked, an institution that buried it, and a price that someone is still paying today. Maybe I am seeing patterns where none exist. Maybe the West made a rational scientific decision in the 1940s, and the Cold War simply delayed a correction that was always going to happen eventually. Maybe the inconsistent early results were reason enough to walk away. The simplest explanation is often correct. But then I look at the Eliava Institute, still operating in Tbilisi, still treating patients, still maintaining phage libraries that Western medicine is now scrambling to access. I look at Tom Patterson, alive, because his wife happened to be an epidemiologist who happened to know where to look. I look at the 1.27 million who did not have a wife who knew where to look. And I think about what else we dismissed because it came from the wrong country. What other treatments are sitting in laboratories and clinics on the other side of a border, working quietly, while we pretend they do not exist because acknowledging them would mean admitting we chose wrong. The Eliava Institute did not close. The phages did not stop working. The bacteria did not wait for us to catch up. The only thing that stopped was our willingness to look. Tell me what you think. Am I seeing patterns? Or is this what it looks like? There is a document from 1962 that changed the rules for how the FDA classifies biological treatments. It did not just affect phage therapy. It affected every non-patentable cure that existed before it was written. That is its own story, and it is worse than you think.